In this video, we will discuss about the comparison between Calnexin and Calreticulum. As we are aware that from our previous videos that Calnexin and Calreticulum, both of them plays an uh, important role for the proper folding of the proteins, and at the same time helps in the glycosylation, especially or glycosylation, particularly in N glycosylation in the endoplasmic reticulum. So let us now understand the structure of calnexin and calreticulum and what are their differences. So first we will understand about this calreticulum. Here on the left side of the figure is the calreticulum structure, and calreticulum is a ER soluble protein, so it is present within the ER, whereas this calnexin is a transmembrane protein, so it is present at the membrane of the endoplasmic reticulum. So in calreticulum, yeah, this is the N terminal and this is the C terminal, and we will start from the N terminal. And from N terminal, we know that there is a signal sequence uh, which directs it towards the endoplasmic reticulum during translation process, and there is a domain. End domain proteins are divided into different section based on the functions. As we have understood, we have uh, known this from the quaternary structure of the proteins, as which is based on dividing separating section of the proteins based on the function that section of the protein does. So this section is divided into N domains, and this section is divided into P domain, and another section is divided into C domain. It is divided into three domains. N domains is here is the uh, we can say three-dimensional structure of the calreticulum. So this end and this is the linear structure of the calreticulum, or we can say just the sequence of amino acids. So in N domains, as it is, we can see that there are two sites at which the cysteine are present. Cysteine is represented by C, which helps in acquiring protein in their native disulfide bond, and here it is represented in the yellow color at C88 and C120. And after that there is a P domain. P domain contains two kind of repeats. One is represented by pink circle, another is represented by the square, and P domain is actually a term. P domain is actually a turn, and this helps in making a bond with ERP57, which is a protein from a PDI family, that is protein disulfide isomerase family, that also helps the protein or the newly co-translated or translated proteins to acquire their native disulfide bond. So this N and P domain together function as a folding unit, or together function as a chaperones. And whereas the C domain of the calreticulum is slightly different from calnexin, and it mainly functions it and this C domain makes this calreticulum as a multi-process molecule. So this C domain will help the cell uh, will help the first it helps as a Ca2 plus buffering protein in the endoplasmic reticulum, or we can say that whenever there is a sudden fluctuation in the calcium requirement within the endoplasmic reticulum, this that will be maintained by the calreticulum. At the same time, it also helps as a signaling. It also helps as a signaling protein to maintain the uh, calcium content of the cells. So that we will discuss about its multi-process function in the some other video. However, we just need to understand at this point of time this that the C domain is also the C domain act as a Ca2 plus buffering. Ca2 plus C domain makes the calreticulum to act as a Ca2 plus buffering protein for endoplasmic reticulum. At the same time, as a signaling, it can also act as a signaling protein to maintain the Ca2 plus level within the cell also. So this is all about the calreticulum structure. Now we will understand some of the structure about calnexin and what are the differences between calnexin and calreticulum. So let us start with the N terminal also. So Calnexin also has this signal sequence which directs it towards uh, endoplasmic reticulum, and it also has this N domain. N domain here the cysteine is present at 161 and 195. That helps to form thiol bonding. That helps to form thiol bonding with the newly co-translated or newly translated protein, and helps them to acquire their native structure, and it is represented by the yellow color at in three-dimensional structure, that is at cysteine at 161 and 195. And after that it also has this P domain, which is like a turn uh, shape, and it has two types of repeats. One is represented by circle, pink circle, and another is represented by pink square. And this is the domain where ERP57 binding occurs. That helps the protein to acquire the native disulfide bond. After that it has this transmembrane domain. This is also the region of P domain, because it again mix with the sorry, this blue circle is also the part of N domain, because it again merges or uh, interact with the N domain. And after that there is an additional domain, that is TM, transmembrane domain. So the transmembrane domain uh, interacts with ER membrane, and all this part is within the ER, that is ER lumen, and this part is outside the ER, we can say in cytosol. So this is the transmembrane domain. It is represented by the green color in linear structure. After that there is a C domain which is present in the cytosol, or the which is present in cytosol, or we can say outside the ER membrane. Here it is represented wrongly. It is not in cytoplasm, it is called cytosol. And one more difference that we need to understand, that since the calreticulum is the ER lumen protein, it has the signature sequence of KDEL. Any protein that has the KDEL sequence will finally has its final destination in the endoplasmic reticulum, whereas as we know that calnexin is a transmembrane uh, RRE, that is not necessarily that this section of the protein will be in the ER lumen. It will remain to the this sequence might be related to, or might helps the calnexin to attach to the to move close to the membrane of endoplasmic reticulum. And so in the next video or in the subsequent video we will discuss about this functioning of this calcium buffering domain of calreticulum. So that's all for this video. Thank you.